Welcome to our 4th annual Gibco 24 Hours of Stem Cells virtual event. Thank you everyone for joining us for the first live presentation of this year's event. Today's presentation will be given by Dr. Rhonda Newman, who joined Thermo Fisher Scientific in 2010 after completing her PhD at the University of Iowa with Madeline Shea and a subsequent postdoc working with Ken Brahota at the University of Oregon. Dr. Newman is now an R&D scientist at Thermo Fisher, where she works on developing next-generation stem cell culture systems. Before we get started, if you'd like to submit a question for Dr. Newman during the presentation, you can submit your question using the green Q&A button at the bottom left of your screen. Questions will be addressed at the end of the presentation. With that, I'd like to hand over the reins to Dr. Newman, and welcome to our live event.
Great. Thanks, Yvonne, for the introduction. So today I'm excited to present to you about our upcoming feeder-free media system, which is a robust media system called StemFlex, and it's really evolving your media system to meet your modern stem cell applications. So with that, first, many of you know that Thermo Fisher offers a variety of stem cell workflow tools, going from genetic characterization of your patient samples, somatic cell reprogramming them into induced pluripotent stem cells, as well as culturing them in feeder-dependent and feeder-free media systems, and subsequently providing a number of characterization tools for assessing maintenance of pluripotency, as well as morphology, and also looking at the engineering workflow with CRISPR-Cas9, being able to edit your cells, and furthermore differentiating them into our neural lineages, definitive endoderm, as well as dopaminergic neuron and cardiomyocytes, and finally measuring them in biochemical and cellular assays. So today I'll be focusing on the media system aspect, but also highlighting some of the ways in which the media system can enable you to work across the workflow, from reprogramming, engineering, as well as downstream differentiation. So as you may know, we currently offer three media systems. So our KSR multi-species supplement, which is a feeder-dependent system. This is on feeders and does require some routine manual cleanup. So it does have a high workflow complexity, as you have to seed your mouse embryonic fibroblasts on attachment factor, condition the media, and seed the cells, and you also have to add the FGF each time. But it is an incredibly robust system, and while the scalability is low, it is a very forgiving system. It does require daily feeds. With our feeder-free media systems, which move researchers from benchtop into translational workflows, we offer two media solutions, our Essential 8 medium solution and our Essential 8 Flex medium. These are feeder-free media systems, and furthermore, they are also xeno-free, which is a really great feature for a media system. Makes it a very lean system for your downstream workflow. Unlike the feeder-dependent culture, you don't have to do routine manual cleanup of your cultures in this system, and we do have a flexible feeding schedule option. So what that means is we have stabilized the FGF component in the media system, and therefore it allows for weekend free feedings or ability to skip up to 3 days of feeding a week. The workflow complexity relative to the KSR multi-species for this is very low, and thus the scalability is very high, and it is rather robust, so it has good robustness for routine culture of your pluripotent stem cells. So now I'm going to introduce to you our StemFlex media system. So this media system is a more robust media system for feeder-free culture of your pluripotent stem cells. It does have animal origin to it, so it does have a bovine serum albumin component to it, but it's lean enough to really minimize the components that may interfere with your optimal downstream workflow applications, in particular single cell passaging, gene editing, as well as reprogramming. It is manufactured as a Gibco CGMP quality standards, and it's offered as a 2-component kit. So it has a 450 ml basal medium and a 50 ml of a 10x supplement, which you add to it. This media system is coming in January of 2017. However, if you're interested in getting a hold of it ahead of that time and evaluating the prototype now, then contact your local sales representative or go to the stem cell research booth after the talk. So a little bit about the StemFlex Media System is it has unprecedented workflow compatibility. So it has a very flexible matrix selection, allowing you to choose from defined matrix conditions such as our recombinant human vitronectin, the truncated form, or the recombinant human laminin 521, or also our HTSC qualified Geltrex matrix. You also have flexibility in your level of dissociation for your downstream applications. So for routine clump cell passaging, you can use Versine solution or a 500 micromolar EDTA solution. If you need to go to a smaller cell cluster, you can use StemPro Accutase to get it down to 2 to 3 cell clusters for high throughput screening, and furthermore you can do single cell passaging using the EE Select enzyme. And I'll be talking to you a little bit about a supplement, RevitaCell supplement, that can be added in combination with this. There is extreme flexibility in the feed schedule in this system. So as I mentioned, you can skip up to 3 days a week. Just a characteristic workflow is shown here at the bottom, where the cells are passaged on day 1, you feed them on day 2, and then you let them rest on day 3, and then you passage on day 4, day 5 double feed, and then you don't feed them over the weekend. So this is one such workflow scheme, but there are additional workflow schemes that you can find online. So now I'm just going to step through some of the pluripotent stem cell workflow challenges and how the StemFlex Media System supports you through these challenges. So the first is looking at culture. So oftentimes you hear of aberrant differentiation due to unstable growth factors. So that's one feature that really is stabilized in the StemFlex Media System and our Essential 8 Flex Media Systems. And so here I just present to you some data that looks at the percentage of FGF concentration relative to time 0. And we basically took media aliquots and put them at 37 degrees, 5% CO2, and pulled out aliquots over varying times. So you can see that time at 37 C, 5% CO2. What you see is very robust stabilization of the FGF component in both the StemFlex and Essential 8 Flex Media Systems, and this is really what supports the cells in order to allow for weekend-free feeding, so up to 72 hours without feeding. Whereas the M-teaser system, you can see that it falls off very quickly, and by 24 hours it's pretty much gone, and then you see the Essential 8 Media System. So what does this mean in terms of the workflow? So we challenged it by looking at the flexible feeding schedule and looked over a number of passages. So here we went through the weekend free feeding schedule, accomplished this for 6 passages, and then looked at the maintenance of the pluripotency marker expression. Here we are looking at OCT4 and NANOG and the StemFlex Media System relative to M-teaser. What you can see is there's a drastic fall off of NANOG's expression and a significant decrease in OCT4 expression over that time frame. Furthermore, if you look at immunocytochemistry images of this using our 4-marker pluripotent stem cell ICC kit, you can see that for the SSEA4 and OCT4 expression, you see maintenance of SSEA4, but you see large holes that have lost OCT4 expression in the M-teaser cultures, and you see very nice morphology maintained in the StemFlex cultures. Looking at the SOX2 and TRA1 to 60, you see still maintenance of the SOX2 expression, but you see gaps in the TRA1 to 60 expression for the M-teaser system. So this is just an example of how the FGF stabilization really allows you a flexible feeding schedule to support your cells long term. So now we're going to move on to a second part of culture that is a workflow challenge. So talking about slow and variable recovery rates post-passaging, which could just be from over-trituration of your cells during passaging time. And this is really linked with poor cell survival following single-cell dissociation even, or into small clusters. But for single-cell passaging, why would you want to go into that workflow pathway? A number of high-throughput screening events in order to make sure that you have consistent cell seeding across the plate. Single cells are important for that step. Also, in a number of downstream differentiation protocols in which the cell seeding density is critical, we found that single-cell passaging of stem cells has been important. And furthermore, delivery of protein or nucleic acid content via electroporation. So here on the bottom, I just show an example single-cell passaging challenge workflow that we used for assessing how the StemFlex Media system performed in this. So we grew the cells to be between 40 and 80% confluent, and then we EEE select dissociated them by treating them for 5 minutes at 37C, and then triturated them with a P1000 pipette tip, diluted them down and spun them down, counted them, and seeded them at 25,000 cells per centimeter squared. And these are incubated at 37 degrees, 5% CO2. And then we looked 24 hours after post-passage at the cell survival. Then we continued to feed daily for M-teaser-1 cultures or every other day for the StemFlex or Essential 8 cultures. And then finally looked at the assessment of the post-passage cell survival once the StemFlex cultures reached passaging confluency. What you can see is that 24 hours post-passage in the absence of ROCK inhibitor, you can see that StemFlex Media System outperforms M-teaser and Essential 8 Flex on all three of the matrices. So looking at Geltrex in the blue bar, recombinant human vitronectin in the red bar, and then recombinant human laminin in the green bar. And I just show a few representative images of what the cells look like post-passaging. 24 hours post-passaging, looking at lower confluency of cultures passaged in M-teaser relative to the StemFlex Media System. Furthermore, when the cells are allowed to go through the first passage and then we look at the population doublings per day, you can see that the StemFlex system again outperforms the M-teaser and Essential 8 Flex Media systems across the three matrices. And so this really indicates that StemFlex Media is providing improved support to stem cells following single-cell passaging. And I'll talk to you about a little bit later about going to an even lower level of seeding at 1 cell per well. So one of the things I mentioned earlier in the talk is that I wanted to talk to you a little bit about an additional component which can provide additional support to your cells. So it's called RevidaCell Supplement, and this was initially developed for post-thaw recovery of pluripotent stem cells, but it can also be used in combination with Essential 8 Medium to enhance single-cell passaging applications. So what the RevidaCell supplement is it's a 100x supplement. It's only added for the first 18 to 24 hours post-passage, and it allows for maximum post-thaw recovery, but also maximum post-single-cell passaging. It's an animal origin-free and chemically defined solution, so that pairs nicely with the Essential 8 Media System, and it contains the ROCK inhibitor as well as antioxidants and free radical scavengers, and this helps support cells during stressful events. So we wanted to see if it made a difference in the StemFlex Media System. So in order to do this, we just did a slight variation of the challenge workflow in which upon the single-cell passaging, we seeded them in the absence and presence of RevidaCell supplement for the first 24 hours. And then we assessed 24-hour post-passage cell survival. And in all three instances of matrices, we saw a significant improvement in the cell survival from the use of RevidaCell supplement. So it can be used in combination with the StemFlex Media System in order to improve efficient single-cell passaging. So moving on to another aspect of the culture workflow challenge is poor cell survival following cryopreservation. So for this, we have a commercially available solution called the PSC Cryopreservation Kit. And this kit was developed for the Essential 8 media system. But as we know that this is a more robust media system, we wanted to see if we could even use a homebrew solution of just StemFlex with 10% DMSO. And so here we use the PSC Cryopreservation Kit, which contains the PSC Cryopreservation Medium as well as the RevidaCell supplement. And what you can see in the leftmost figure is the percentage confluency 24 hours post-thaw versus the Cryo Medium in Recovery supplement. So what you can see is that we see very similar performance of the ready-to-use PSC Cryo Medium in comparison to the homebrew StemFlex plus 10% DMSO. We do still see an improvement in cell survival upon the addition of RevidaCell supplement. And you can see a few representative images here on the right showing this is without a media change, so you can see that many of the cells survived and stuck very nicely to the plate. So this is a very nice, robust media system for your pluripotent stem cells. So the final aspect of the culture workflow challenges is the challenging workflow schemes that are required to transition your cells from richer media systems. Oftentimes groups will get in cells that have been in feeder-dependent contexts or they've been in other media systems such as MTeaser. So we wanted to have proof of principle that you could easily transition the cells over into the StemFlex media system. So here we took a KSR feeder-dependent medium system that was an established cell line in culture, so it's in culture at this time, and then we collagenase passaged it for the panel shown below, but you could also use disk-based passaging. And we seeded those into either the StemFlex GelTrex system or StemFlex onto vitronectin. And that was carried out for one passage, and then finally just performed versine clump cell passaging. And so on the bottom left-hand panel, you can see following that versine clump cell passaging, you can see very nice recovery on the vitronectin as well as the GelTrex substrates. Furthermore, assessing pluripotency using OCT4, which we found to be very sensitive to maintenance of pluripotency, you can see very nice morphology and maintenance of that pluripotency over this transition. So a rather simple workflow for transitioning your cells over into this system. For the MTeaser Matrigel established cell line in culture, all you simply had to do is perform versine or 500 micromolar EDTA clump passaging, and then this can be seeded directly into the StemFlex GelTrex or the recombinant human vitronectin system. And so on the bottom left-hand panel, you can see the percentage confluency over the time post-passage and post-versine passage, and you can see that the StemFlex system recovers very nicely, whereas there's a little lag in the MTeaser system. But what you can see is that on both the recombinant human vitronectin as well as the GelTrex substrates, you can see very nice maintenance of pluripotency in both systems. And I should mention that we recommend 2-passage adaptation for a full adaptation into the media system, a minimum of 2 passages. So now we're going to move on to the reprogramming workflows and some workflow challenges for this. So some of the things that we've heard is that customers have challenges with small size of colonies, so this makes it challenging for positive selection as well as the easy propagation of their cultures. Also the requirement of daily feeding upon addition of the PSC medium. So I'll go through the workflows and just show you how many days you would end up having to go for reprogramming of your cultures. So looking at the workflow, first we looked at the human dermal fibroblasts. For this Cytotune 2.0 workflow, basically on day 0 you perform your transduction of your fibroblasts, and then you just replace the media up to day 7, and then they're transitioned over onto a GelTrex substrate, or we transfer them over to MEFs in the context of the KSR feeder-dependent system. And then finally on day 8, we switch over into our iPSC medium. And from there, the M-teaser cultures we fed daily, the KSR-based cultures, or essentially RUO cultures, we fed daily, and then the essential 8 Flex and the StemFlex media systems we just fed every other day. And this goes up to day 21, and then we were assessing how well they did as far as reprogramming efficiency using alkaline phosphatase staining. So for looking at the morphology of the cultures, so here we show a few representative images of just one of the donors that we assessed for the fibroblast reprogramming. And you can see the KSR multi-species medium has a very robust reprogramming efficiency. So this is on feeders, and so it does have a higher workflow complexity downstream, and it's daily fed. The essential 8 Flex culture, you can see, well, it has very high reprogramming efficiency. Many of the colonies are smaller. And then you can see with the StemFlex media system on gel tracks where they're fed every other day, you see many large colonies that are easy to select. And you can see a representative image there on the left. And for the M-teaser gel tracks fed daily, again, you can see some large colony size. So next is poor reprogramming efficiency, and so here we looked at a variety of cell sources as well as donors. And so here we looked at the Cytotune 2.0 reprogramming workflow, and we used a combination of 3 donors. So donor 1 and 2 are both adult human dermal fibroblasts, and donor number 3 is a neonatal human dermafibroblast, and both of the, in case you're wondering, donor 1 and 2 were both from female tissues. So in looking at the reprogramming efficiency across the different media systems, you can see very comparable reprogramming efficiencies between the essential 8 media system, which was fed daily, and the essential 8 Flex media system, which is fed every other day. You can see a decrease in reprogramming efficiency for M-teaser, and StemFlex is in the middle. So it does have improved reprogramming efficiency over M-teaser. And on the leftmost panel shows on vitronectin substrate, whereas the rightmost panel shows the reprogramming efficiency on the gel track substrate. So very comparable reprogramming efficiency is seen on both substrates. And it's nice that there is that requirement for only the every other day feeds for both the StemFlex and essential 8 Flex media systems. So we also wanted to look a little bit at the blood cell reprogramming workflow. So we use Cytotune 2.0 to reprogram in CD34 positive blood cell reprogramming challenge workflow. So here we went through the Cytotune 2.0 reprogramming workflow, and then starting on day 8 we switched to the PSC medium and fed them again daily or every other day on the every other day feed schedule. This particular donor was very robust, so by day 14 it was already ready. The colonies had gotten large, so by day 14 we had to perform alkaline phosphatase staining, but it's a little out of the norm. But looking at the reprogramming efficiency of the relative media systems, you can see that for essential 8 Flex and StemFlex, they actually behave very similarly for this media system on both the recombinant human vitronectin as well as the gel track substrates, and we did see improved performance over the M-teaser 1 system for this workflow. So now that we've talked a little bit about the culture as well as the reprogramming features, now we want to move on to the engineering. So this is something we're going to have we have a live chat session tomorrow, so if you have additional gene editing informational questions, then please feel free to join that informational session, as we have our gene editing experts on that call, on that live chat, which can provide you with additional information. So one of the first challenges in the PSC workflow for engineering is that there's a challenge of poor cell survival post-edit and even aberrant differentiation upon expansion post-editing due to the stress of the cells during that event. So as you may know, we offer a number of workflow solution tools for CRISPR-Cas9 gene editing, and these allow for delivery of content in various formats. So we offer lipofectamine 3000 for CRISPR DNA vector delivery. We have lipofectamine CRISPR Max, and this can be used for Cas9 protein complex delivery. And then we have the lipofectamine messenger Max, and this can be used for delivery of mRNA and guide RNA mix. But the neon electroporation device actually supports all of these formats, and that's what we used in this workflow challenge. So along the top, I just showed the CRISPR-Cas9 gene editing challenge workflow. So here we took feeder-free iPSCs, we singularized them using EE select, and then performed genome editing using the neon electroporation device of our CRISPR-Cas9 protein and the HPRT guide RNA, which is a control guide RNA that you can order through us to optimize your system. These were plated and analyzed for post-transfection efficiency. So on the bottom left-hand panel, I just showed recovery in the absence of Revital supplement. These are seeded very high. It's actually 100,000 cells for a 24-well plate. So they're seeded at very high cell seeding density. You can see the StemFlex media system recovers very nicely. And then you can see that's followed by essential 8-flex and then M-teaser. Something I want to point out between the left and the right panels is that if you include Revital supplement for the first 24 hours, there's no benefit to the StemFlex media system at this cell seeding density, but there is a significant benefit for inclusion for the essential 8 media system, and the M-teaser media system, there's no advantage. So now I'm going to ask Tracy to just pull up a movie. So I want to show you this movie to show you how the morphology changes over time and how the cells progress. So Tracy, can you pull up that video? The suspension. So if you come back to your media, your culture system, and you see the floating cells, it's very normal for a feeder-free media system, so don't be alarmed. And that morphology does resolve, so you see very nice maintenance of that pluripotent morphology. The next thing we wanted to look at was poor clonal cell survival post-edit. So through the gene editing workflow, in general, what we are doing is delivering the content and then screening for the editing efficiency. So that can be done via a number of methods depending on what you've delivered. Some people use flow sorting. So in-house we use a TRA1 to 60 stain for looking at pluripotent stem cells and a live cell or a dead cell marker, propitium iodide, to make sure that we're not sorting dead cells. And then these were, so this workflow scheme, we basically take the feeder-free iPSCs, we singularize them using triple select, and we flow sort them on the live pluripotent stem cells. These are then seeded at very low cell seeding densities into 96 well plates, so at 1, 3, and 5 cells per well. And we use our recombinant human laminin 521 in this particular workflow because it is the most robust matrix system for pluripotent stem cells. And then following that seeding event, the cells are fed every 3 days, and finally we assess the confluency 14 days post-seeding. So on this slide, we show the percentage of wells that have clonal expansion. On the leftmost panel, this is seeding in the absence of ROCK inhibitor or Revital supplement on that recombinant human laminin 521. You can see that the StemFlex system does perform the best in terms of recovery. So you can see up to 50% of your wells recovering if you're plating at 5 cells per well. So that's very, very great very great clonal expansion. It allows for very efficient screening of your clones. On the right-hand side, we show that in the presence of Revital supplement on the laminin substrate, you can see that there is a further boost in the cell survival. So you're able to get up around a little more than 20% of your clones being recovered from seeding of a single cell per well. So it's a very nice workflow tool and indicates the robustness of this media system. So finally, we're looking at the differentiation workflow. So here we're looking at the aberrant differentiation upon expansion of the pluripotent stem cells. And so we just wanted to make sure that that wasn't occurring in this media system. So here we look at maintenance of the normal PSC properties, and we looked at normal pluripotent morphology and karyotype, as well as expression of pluripotent stem cell markers over 20 plus passages. So here we show very nice maintenance of normal pluripotent morphology with nice smooth edges, and a closer zoom in of the cells would show a high nuclear to cytoplasmic ratio. We did send these for karyotyping and show normal karyotype come back up to 20 passages. And furthermore, looking at it using our 4-marker pluripotency ICC kit, we see maintenance of pluripotency markers, the SSCA4, OX4, and then on this slide showing the SOX2 on trial 1 to 60. So we have shown up to that, up to 20 passages, and these studies are continuing, so we will have this continue out to up to 50 passages. So finally, we come to incompatibility with downstream differentiation workflows. So as you may know, Gibco offers a number of differentiation kits, downstream differentiation kits. And so I'll talk to you a little bit about each of these kits and their features, and then also share with you how they worked with the StemFlex media system. So for the first, looking at maintenance of ectoderm differentiation potential, we use the PSC neural induction medium, and we also use the human neural stem cell ICC kit to assess how they're doing. So here we have the Gibco, basically it comes as a basal media and supplement. And what you can see is for the StemFlex media system, you can see very nice expression of nestin and SOX1, which are both markers of neural stem cells. And so this is showing compatibility of the StemFlex media system with the PSC neural induction medium, and also that we maintain the ectoderm differentiation potential of the system. Then we move on to our Gibco PSC definitive endoderm induction kit. So this is a 2-part kit which gets you to definitive endoderm progenitors. And so it basically has a medium A that's added on one day, medium B that's added on the second day. And we assess the differentiation potential of these by looking at expression of CXCR4 and downregulation of PDGFR alpha. And so what you can see is that you can get up to 99% expression. So now we're going to move on to the Gibco PSC cardiomyocyte differentiation kit. So this particular kit comes as a 3-component kit. It has a medium A, medium B, and a cardiomyocyte maintenance medium. This allows for very nice expression of cardiomyocytes, and so here we just show on the bottom panel the TNNT2 expression day 11 post-induction. And I'm going to ask Tracy to pull up maybe for me now, looking at the cultures beating on day 9. So they actually beat as early as day 8. Tracy, can you pull up that video for me? Pattern on this on the cardiomyocytes, so showing very nice expression of the cardiomyocyte We also paired this with our human cardiomyocyte immunocytochemistry kit, and so this allowed us to look at an early mesoderm marker in KX2.1 and our troponin T2. And you can see very nice expression of both in this media system, and this was assessed day 19 post-induction. So you can keep these going for a long term. So now that brings us to looking at our where we are with the PSC culture media systems and today, and looking at how we would position each of these media systems, as they all have their strengths. So we have the KSR multi-species supplement, the StemFlex Medium, which is coming in January of 2017, and finally our Essential 8 media systems. So I just went through and tried to scale them based on their robustness. We do know that the feeder-dependent system is incredibly robust. It allows you to keep your cells in culture. You could accidentally overgrow them slightly and not have detriment. With the StemFlex media system, you can grow them to high confluency, but you wouldn't want to leave them there for a long period of time, so I listed that one as better. And then the Essential 8 media system is a leaner media system, and therefore isn't quite as robust as what we see for the StemFlex media system. For the somatic cell reprogramming, we do see that the KSR multi-species supplement is the best for reprogramming, followed by the Essential 8 media system and Essential 8 Flex. They have good reprogramming efficiency, but the StemFlex media system does show strength in this area and provides very nice, robust sized colonies. So finally, that brings us to our gene editing support. So we explored a few parts of the workflow, looking at the CRISPR-Cas9 delivery via electroporation and the cell survival from that. And then we also looked at clonal expansion following flow sorting. And so for this, we listed the StemFlex media as the best system for this particular instance. So with that, I'd like to thank all the contributors. We had a number of contributors internally from Thermo Fisher Scientific, the project team. Eric Willems he helped out a lot with the gene editing workflow pieces and works in our custom biology team in Carlsbad, California, has a lot of insight into that. And I'd invite you to join his live chat session tomorrow if you're particularly interested in gene editing applications. Lawrence Angenero, who helped out immensely with this project. Renee Quintanola provided a number of the reprogramming experiments actually conducted all the reprogramming experiments in this particular presentation. Rex, who also helped out Eric and all the rest of the team. And we'd also like to give a special thanks to collaborators at CCRM and Harvard for their assistance in selecting our final formulation. So with that, I'm open for any questions.
Thank you very much, Rhonda, for that excellent presentation. We are now going to open the floor for questions. As a reminder, if you'd like to ask Dr. Newman a question about her presentation, please use the green Q&A button at the bottom left of your screen. We have had a number of presentations coming in while Rhonda was speaking, so we're going to kick it off with those. The first question was whether EEE should be pre-warmed before adding to the cell.
That's a very good question. So in general, we keep our EEE at room temperature. You can pre-warm it, but you don't need to. So it does need to be at least at room temperature. Obviously, if you do pre-warm it, it can act a little faster. So in general, what we recommend is using room temperature EEE select for 5 minutes at 37 C, 5% CO2.
Fantastic. The next question is, how stable is the new StemFlex media? Can it be aliquoted and refrozen for prolonged use?
Yeah, so that's a very good question. So StemFlex media is very stable. We've evaluated stability of the media up to 2 weeks at 4 degrees C, so you can prepare your 1X media, keep it at 4 C for up to 2 weeks. If you find that you wouldn't need the full amount of media in that 14-day period, then you have a couple of options. So the first would be that you aliquot and refreeze the supplement. We don't recommend doing additional freeze thaws after that, though, so keep in mind that you'll need to figure out what aliquot size you need. And then the second option that you have is to reconstitute it as a 1X media, and then that can be aliquoted away into and frozen at -20 for up to 6 months.
Great. We've also had a question. You mentioned the sorting during the gene editing process. A question was whether the same kind of staining and flow cytometry can be used to actually sort poor quality and high quality iPSCs and isolate them that way.
Yeah, so there's a number of ways. I mean, you can definitely, I've had a number of collaborators who are using their gene delivery context using a GFP reporter so that they know that they've gotten it into their cells, and then they flow sort based not only on the TRA1 to 60 expression, but then they can also flow sort on the propydium iodide to make sure that they have live cells or make sure it's propydium iodide negative, and then also look at their GFP expression to say that they've truly been delivered the content that they were needing to be delivered. So that allows you to sort away the ones that aren't potentially carrying the target of interest that you're interested in.
Fantastic. And can the same TRA160 stain also be used for sorting out high-quality pluripotent stem cells and other workflows?
Yeah, so TRA1 to 60 is a really common one that we use internally. So yeah, we usually pair GFP, TRA1 to 60, and propydium iodide.
Beautiful. We have another question here. Can you readily switch back to Essential 8 for routine culture after using StemFlex for gene editing or flow sorting?
That's a really good question. We've not evaluated that yet. However, I don't see that that would be an issue if you did only want to use it for a specific part of the workflow. However, we have shown that the StemFlex media system does work across the entire workflow. So depending on if you wanted to move into a translational system, then obviously moving to Essential 8 would make a lot of sense. So so I believe it's possible, and we'll have to evaluate it.
Mm-hmm. We're getting a lot of questions about gene editing. This is clearly a very hot topic, especially with this new StemFlex medium. The next question is, do you have any protocols you can share for gene editing PSCs in StemFlex?
Yeah, so we're working on putting together a protocol for the delivery using our neon electroporator and following up, going through the flow sorting and clonal expansion aspect of the cultures. So that will be available at launch. Furthermore, we're looking at the CRISPR Max Cas9 transfection reagent, and we'll be providing guidance on that as well soon thereafter.
Great. What matrix do you recommend to use with StemFlex?
So the matrix depends on what your downstream application of interest is. So in general, we recommend Geltrex medium or Geltrex matrix or HESC qualified Geltrex matrix for routine propagation of cells. And we found that recombinant human laminin is the best in going into our downstream gene editing workflows. It is the most robust.
Can you comment on whether StemFlex can also be used with other cell types like murine pluripotent stem cells?
That's a very good question. We are getting a lot of questions about that, so I'm sure we will be evaluating it shortly. We've not evaluated it yet.
When preparing the medium, should it be filtered? And if yes, how do you recommend doing that?
So it doesn't need to be filtered. So they are manufactured under CGMP sterile requirements. So you don't need to filter it. But if you did want to filter it, you can buy. We do offer now gene filter units that have 0.2 micron filters. But there's no need to filter it.
We have a question about the different methods of packaging for the different media systems that you described. So for the different media systems, what method of packaging do you recommend, single cell or manual? So I believe with manual they mean clump cell packaging.
So for the manual, so for the KSR multi-species supplement, you do have a number of options. So you can either do manual packaging, so you can either score it by hand or in transfer. If you're doing positive selection, you can also use the easy roller passage tool, which we also offer, that very nicely cuts it into the correct sizes. You also have the option of using collagenase or disc space in the KSR multi-species format. And in that particular format, you treat it with a collagenase or disc space. They curl up slightly on the side, and then you triturate them into smaller pieces. So that's for the KSR multi-species. For both of the essentially in the StemFlex media systems, we in general recommend versine for continued propagation of the cells, and then for going into single cell passaging applications, we recommend triple E select.
Great. Do we have any indication about using StemFlex for adult stem cells?
For adult stem cells? So
I think the question would be whether it can be used for neural stem cells, hematopoietic, or any other kind of adult stem cell.
Oh, okay. Yeah, so we've not evaluated it in the other other adult stem cell contexts.
Mm-hmm. We don't have a flow sorter. What seeding density would you recommend for clonal expansion for downstream colony screening?
Yeah, we've had a number of our collaborators that are kind of working along those lines that they don't have the flow sorter. And so in that instance, they're seeding between 100 and 300 cells per centimeter squared. And this allows for really nice clonal expansion of the colonies, and then they're able to easily go in, and you can manually score and transfer those over.
Great. What are my other flexible feeding options? Is it okay to feed daily, and when would it be recommended?
That's a very good question. So if you go to our Essentially Flex Medium workflow page, you can see there's a number of workflow schemes that are laid out for the Flex systems. And you can see that it's incredibly flexible and allows you to skip up to 3 days a week. As far as daily feeding, we do see that there is a benefit if you're going into a very strenuous situation, such as going through these gene editing applications. We do see a benefit to the daily feeding of the stem cells ahead of that workflow. But in general, you don't need to. But it would not harm the cells at all if you fed daily.
Wonderful. Why would I use StemFlex instead of Essential 8?
So if you're really having a hard time with your media system, we've had a number of people that have approached us and saying that they have disease lines that are particularly challenging, and they're working on it basically for a disease modeling perspective, so more of a research aspect. Then StemFlex provides a very nice, robust media that allows them to support a very robust reprogramming expansion and gene editing workflow for that type of person. Moving into the translational clinical space, that you're going to want to move away from bovine serum albumin, and that's where Essential 8 media systems come into play, moving more to the translational space. They're very good media systems, and we do have we do have additional things that can be added to help support them through different parts of the workflow. So one I mentioned was Revital supplement. The other is laminin 521. So both of those things in conjunction with the media system really help with the leanness of that media system.
Wonderful. We have a question about characterization. So you are using karyotype to check your cells after multiple passages. Do you think that NGS or next generation sequencing analysis could also be of benefit?
Yeah. So we actually have a number of collaborators that are going to be doing that with the cells as well, and that's definitely on our radar to do next. And we routinely check the karyotype. We generally will check it every 10 passages, and we bank away cells every 10 passages just in the event that there's ever an issue. That's just in general, our general workflow for stem cell culture.
We had a question, when will this product be available and how can I get access to a sample?
So the product will be available mid-January 2017. However, if you would be interested in assessing it now, we do have a prototype which you can assess if you talk to your local sales representative. Alternatively, as I mentioned during the presentation, you can reach out to the stem cell research booth that's offered in the 24 hours of stem cells.
What is the half-life of StemFlex Medium? I can hear the referring to the half-life of FGF2.
I'm sorry, you're breaking up a little.
The question was, what is the half-life of StemFlex Medium? I believe they're referring to FGF2.
Okay, I'm sorry, I still couldn't get that. One more time.
What is the half-life of StemFlex Medium?
Oh, what is the half-life of the StemFlex media? So as far as, so we've not looked to see how long it can go for sure. You mean so I'm assuming that you're meaning if you're to put it in a 37C 5% CO2 incubator, how long will it take for it to fully decay out? And we've not assessed out how far that goes yet.
Okay. So what is the longest amount of time, I guess, taking that question, that we recommend skipping a feeding?
So the longest amount of time that we do recommend is up to 72 hours.
Great. Another question, is it possible to reprogram cells using Essential 8 and then transition them back into StemFlex Medium upon harvesting the IPS line?
Yes, so we have some collaborators that have been working on using either Essential 7, so having the Essential 6 and bolting on the BFGF, and looking at reprogramming in that workflow system, which is very, very efficient, and then transitioning those over into the StemFlex media system, and it works very well for them.
Beautiful. Do you have any tips for successful clonal expansion post-slow sorting, such as adaptation onto the recombinant human laminin 521 matrix or changing the feed schedule?
Okay. So for that particular workflow, what we found, a few tips that we found to be really helpful is, again, as I mentioned, the daily feeding ahead of that of the downstream transfection or the upstream transfection workflow. In addition, daily feeding during the expansion post that time does seem to have a benefit. And then we found that if you go through, if you're using the laminin 521 substrate, you see even greater benefit if you fully adapt the cells over onto it. So if you adapt them over for 3 passages on the recombinant human laminin StemFlex matrix, media matrix combination, then we find the best cell survival.
Beautiful. Along those lines, what cell seeding densities are recommended for routine passaging of PSCs as single cells? And are there any precautions I could take to make sure that they remain karyotypically stable?
Yeah, so in general, what we recommend is that if you're going to need to passage them every 4 to 5 days and you're including Revital supplement, that you passage them at a cell seeding density of 12,500 cells, viable cells per centimeter squared. Alternatively, if you're working in the absence of Revital supplement, then 20,000 cells per centimeter squared. And again, we found that really for best support of your pluripotent stem cells, if you're doing a routine passaging as single cells, then we found that the laminin substrate does provide the best support.
Wonderful. We have a question. How does this medium compare to M-teaser?
That's a very good question. So basically, whenever we sought to develop this system, we just reviewed the currently published media formulations that are out there, and we really worked to highly streamline and optimize the system. So we wanted to make sure that our concentrations were optimized of different components. We were able to significantly strip out a number of components that we found to be detrimental to the across the PSC workflow. And so from that, we end up with a very streamlined system relative to M-teaser.
Beautiful. Well, with that, I think we are going to be ready to close our session. We did have a question about accessing your presentation after the fact. So this is going to be available on demand following this live event. It's going to be available in this 24 hours virtual system for 12 months through next October. So for anyone interested in sharing this with your colleagues or getting back and reviewing this information, you'll be able to do so at your leisure. So thank you, everyone, and Dr. Newman for spending time with us today. This presentation does count towards your event certification, so please click on the event certification button in the bottom left of your screen in the virtual environment to find out more about certification and how to get your free t-shirts. And if you do have some extra time, please be sure to check out our stem cell research booth in the exhibit hall and ask about sampling the new StemFlex Medium. We do have experts standing by who would love to hear from you, and we'll be happy to address any additional questions you may have. So with that, thank you again, Dr. Newman, and thank you to the audience. And we will be closing this live event.